So if you were to sit down right now and just start reading your own DNA out loud, like 1 letter per second, 24 hours a day, do you know how long it would take you to finish?
I mean, I know the math, but it's still staggering to think about.
Yeah, it would take exactly 1 century, 100 years of nonstop reading. And, for decades, the foundational assumption of modern science was really that if you actually read that century-long book, it would be, well, mostly just evolutionary gibberish.
Right, exactly. A lot of noise.
Yeah, noise. We thought we would basically find this biological attic, just a messy, chaotic space stuffed with random genetic typos, half-finished experiments, and just the accumulated junk of billions of years of random trial and error.
Which was a perfectly reasonable hypothesis at the time, given the models we were working with.
Sure, but we were entirely wrong. I mean, you are not carrying around a diary of biological accidents. You're carrying around this mathematical masterpiece.
It really is, without exaggeration, one of the most profound paradigm shifts in the history of science. I mean, the sequencing of the human genome didn't just give us a map of our biology. It completely upended our philosophical understanding of what biology actually is.
Yeah.
We went looking for the residue of blind chance, like you said, and instead we found an architecture that is so precise, so deeply integrated, that it's literally forcing us to rethink the origins of life itself.
And that tension right there is exactly our mission for today's deep dive. We're going to explore a highly provocative new academic paper by Jamie Thornberry. It's titled The Ontology of Life as Ordered Light. And I'm going to read the subtitle here. Artificial Intelligence, Genomic Intelligibility, and the Limits of Evolutionary Explanation.
It's a heavy title, but the implications they're massive.
So massive. We're going to dive into what happens when cutting-edge artificial intelligence basically collides with the human genome. Because Thornberry argues that our ability to describe what the genome does is, ironically, making it infinitely harder to explain how it got there using standard scientific models.
Yeah, the central premise here is this widening gap between descriptive success and explanatory sufficiency.
Right.
Thanks to computational models, we have never been better at describing the mechanics of life. But Thornberry points out that the more clearly we see this astonishing rule-governed structure of the genome, well, the less adequate our classical, undirected evolutionary mechanisms become at explaining its absolute origin.
Okay, let's unpack this because we really need to set the stage to understand why this was such a massive shock to the system. Let's go back to the 1990s, right? The era of the Human Genome Project.
Right, the Wild West of genomics.
Exactly. We're dealing with 3.2 billion base pairs. Back then, what was the actual expectation of what we were going to find in there?
Well, the prevailing scientific and really the metaphysical framework was built heavily on this concept of stochastic accumulation.
Let me stop you right there. Stochastic accumulation.
What?
Is that basically just the academic way of saying nature threw genetic spaghetti at the wall for couple billion years just to see what stuck?
That is a highly colloquial but remarkably accurate translation, yes.
Okay, good. Just making sure.
Stochastic essentially means having a random probability distribution. It's a pattern that you can analyze statistically, but you can't predict it precisely. So the assumption was that mutations happen randomly, right?
Right.
Most do nothing, some are harmful and get weeded out by natural selection, and then a tiny, tiny fraction are beneficial and get kept. So over billions of years, this trial and error process builds a human.
So the expectation was that the genome should basically look like a patchwork quilt of all that chaotic history.
Exactly. We expected to find these small islands of functional codes surrounded by vast, vast oceans of meaningless evolutionary noise. That's what was famously dubbed junk DNA back in the 1970s.
Right, because if a process doesn't plan ahead, it doesn't organize. It just kind of piles things up.
It just accumulates. But what emerged from the sequencing, and especially from subsequent deep analysis initiatives like the ENCODE project, it completely contradicted that expectation. Biologists didn't find accumulated disorder refined by selection.
They found something else entirely.
They found layered regulation. They found what we call long-range genomic dependency. They discovered functional precision at scales that simply resist reduction to random, unguided accumulation. What's fascinating here is that the genome behaves as a tightly integrated architecture, not an aggregation of independent parts.
The whole biological attic analogy was completely wrong. It's more like, okay, imagine expecting to walk into a hoarder's house, right? Where things are just tossed wherever there's an empty space. But instead, you walk into a perfectly cataloged, highly restricted, climate-controlled reference library.
That's a great way to picture it.
And the location of every single item is dictated by a master organizing principle.
Yeah.
I mean, if you walk into a room like that, that fundamentally changes how you view the builder. Right.
It fundamentally changes how you view the nature of the space itself. I mean, in a hoarder's attic, a box is in the corner simply because there was empty space there when the hoarder walked in. There's no overarching rule.
It's just random placement.
Exactly. But in a reference library, a book's location on the third floor is fundamentally logically related to the location of a book on the first floor. And that is exactly what we found in the DNA. It isn't just a linear string of independent genes doing their own thing.
Which completely breaks my brain, honestly. Like, how does a chemical sequence exhibit that kind of architecture? Because, we throw around the phrase genetic code all the time. It's literally on the cover of every high school biology textbook.
Well, constantly. It's a huge buzzword.
But Thornberry's paper seems to be arguing that we need to take the word code completely literally.
He does. And this is a crucial pivot in his argument. Thornberry insists that biological information is an ontological reality. It functions as a true symbolic code, not merely a series of chemical reactions.
Hold on, before we go any further into that, people use the word ontology to sound smart at dinner parties. But what does it actually mean in the physical context of a living cell? Are we talking about what a cell is made of or what a cell actually means?
That is the perfect question to ask right now, because ontology is the philosophical study of being. It asks, what is the fundamental nature of reality? What categories of things actually exist?
Okay.
So if you are a strict materialist, your ontology says that only physical matter and physical forces exist. Period.
Nothing else.
Right. So in that view, a cell is nothing more than a localized collection of carbon, hydrogen, and oxygen reacting according to the blind laws of thermodynamics.
So just a really, really complicated baking soda and vinegar volcano.
Exactly. Just chemistry playing out. But Thornberry is arguing for a totally different ontology. He's arguing that information and meaning are just as fundamentally real as carbon and hydrogen.
Wow. Okay.
He's saying that the genome isn't just matter reacting, it is matter establishing symbolic reference. And to understand why, we really have to look intimately at the mechanics of DNA.
Well, let's do it. Let's look at the actual molecular machinery. You have DNA, which is made up of nucleotide bases, adenine, thymine, cytosine, and guanine.
Right. A, T, C, G.
Famous letters. And these letters group together in triplets, which we call codons. And each codon corresponds to a specific amino acid. Then those amino acids get chained together, they fold up into a 3D shape, and boom, you have a functional protein that does the physical work of keeping you alive.
That is the mechanical description, yes.
Right.
But here's the critical philosophical distinction that Thornberry highlights. The mapping between those codons and the amino acids they produce is not exhaustively determined by chemical necessity.
Wait, make sure I'm really understanding this. You're saying the physical chemical properties of the DNA letters don't chemically force the creation of the specific amino acid.
That's exactly what I'm saying.
They don't just magnetically snap together because of their molecular weight or their charge.
Precisely. There is no direct, inevitable physicochemical reaction that forces the codon sequence CGT to bond with the amino acid arginine. In fact, they don't interact directly at all.
They don't touch?
No. The translation requires a massive intermediary apparatus, the ribosome and transfer RNA, to read the sequence and fetch the corresponding part.
So, to use an analogy, it's like ink and paper.
Yeah.
The laws of chemistry and physics dictate how the ink adheres to the paper, right?
Yes.
They explain the capillary action, the drying time, all the molecular bonding. But the laws of chemistry do absolutely nothing to explain why the ink is arranged to spell the word APLE instead of the word orange.
That's a perfect analogy.
The chemistry is entirely indifferent to the message being conveyed. The chemistry is just a medium.
That is exactly right. The arrangement of the letters is symbolic. They stand in for a concept outside themselves. And Thornberry argues that meaning, in a scientific and ontological sense, requires three distinct phenomena to be present.
Okay, what are they?
Stable correspondence, rule-governed translation, and functional consequence.
Let's map those three directly onto the human genome so we can actually see the mechanism in action.
Sure. First, stable correspondence. The sequence CGT consistently corresponds to arginine across almost all living organisms. That is a stable, reliable map.
Okay, got it. What's next?
Second, rule-governed translation. We have the transfer RNA, or tRNA, and the ribosome. The tRNA has an anticodon on one end that physically recognizes the DNA sequence, and it carries this specific amino acid on the other end.
So it's literally a physical bridge.
Exactly.
Yeah.
And the ribosome acts as the physical translator, coordinating this interaction based on a strict pre-existing set of rules.
And the third requirement, functional consequence. What does that mean for the cell?
Functional consequence means the symbolic translation actually matters to the physical world. If the translation follows the rules perfectly, the amino acids chain together correctly, the protein folds into the exact necessary three-dimensional shape, and the cell successfully metabolizes energy or, repairs a membrane.
And if it doesn't?
If the translation is wrong, if the rule is broken, the protein misfolds, the function is lost, and the organism might suffer systemic failure.
So because the connection between the DNA sequence and the protein it builds isn't forced by basic chemistry, there has to be an abstract symbolic rule connecting them.
Yes.
I mean, that fundamentally shifts how I understand my own body. We aren't just a bag of inevitable chemical chain reactions. We are the physical manifestation of a symbolic language.
We are materialized information, and this distinction between matter interacting causally and matter establishing symbolic reference, it really cannot be overstated.
Yeah.
When matter interacts causally, like an asteroid hitting a planet, it's just physics. But when matter operates referentially, it implies a formal organization. It operates within a system of signs.
But if this massive 3.2 billion letter code isn't just a blind chemical chain reaction cascading forward, how do the different parts of it actually communicate with each other? We touched on the library analogy and these long-range dependencies, but I want to know how that physically works.
It's incredible when you actually look at it.
Because if a single molecule in my DNA somehow knows what's happening 100,000 base pairs away without a central nervous system, how on earth is it doing that?
To understand this, we have to completely abandon the classic textbook image of DNA as a straight, rigid ladder.
The double helix we all drew in middle school.
Right. Throw that image out. The genome is profoundly, incredibly context-dependent, and its physical three-dimensional structure is the key to this communication. We have regions of DNA called regulatory elements, specifically enhancers and silencers.
And what do they do?
Their entire job is to turn other genes on or off, or to turn their expression up or down like a volume dial.
Okay, so a regulatory element acts like a switch for a gene. But you mentioned these are long range. How far away are we talking?
We are talking about hundreds of thousands, sometimes millions of base pairs away along the linear strand of DNA.
Wait, how does a switch turn on a light bulb if it's a million chemical letters away and there are no wires connecting them?
Because the genome folds. You have about 2 meters of DNA packed into a microscopic cell nucleus.
2 meters in a single nucleus.
Yes. And it achieves this through a highly precise, dynamic, three-dimensional origami. The linear strand loops and bends so that an enhancer region, which might be linearly a million base pairs away, physically folds over to directly touch the promoter region of the target gene.
Oh, wow. So distance in a straight line doesn't matter because it bends back onto itself.
Exactly. We call this chromatin looping, and it occurs within highly organized structures called topologically associating domains.
That is staggering. So the code is written on a one-dimensional string, but it only functions because it folds into a three-dimensional shape, where the exact physical proximity of distant parts determines whether a gene is read or ignored.
Yes, and the timing of this folding is exquisite. Let's look at a concrete mechanistic example, the p53 tumor suppressor gene.
I've heard of that. It's super important for cancer research, right?
It is often called the guardian of the genome. When a cell's DNA is damaged, p53 is activated. But p53 doesn't just do one thing. It binds to thousands of different regulatory sites across the entire genome simultaneously.
Thousands. At the same time.
Yes. It pauses the cell cycle so the cell stops dividing. It activates DNA repair genes. And if the damage is too severe, it triggers apoptosis-programmed cell death to prevent the cell from becoming cancerous.
So p53 is essentially a master conductor.
Precisely. Now imagine the regulatory elements of the genome are like a vast, complex symphony orchestra. You have 100 musicians. The violins are transcribing a specific protein, but their timing, their volume, and their physiological resonance are entirely dependent on what the cellos are doing across the stage and what the p53 conductor is signaling from the podium.
And if one musician plays out of tune,
if one nucleotide out of 3.2 billion is swapped, a single point mutation in a critical regulatory region, It might subtly change how the DNA folds.
Just a tiny bend in the wrong direction.
Right, and that slight fold alteration might prevent an enhancer from reaching its target gene. The gene doesn't turn on, a vital protein is missing. The cell cycle isn't paused, the damage isn't repaired, and that single missing interaction can cascade into massive systemic dysfunction. It can lead to cancer.
Now, if I'm playing devil's advocate here, I might hear that and say, well, that just means the biological system is incredibly fragile. It's a poorly designed system if changing one tiny letter out of billions breaks the whole thing.
It is a really common misconception to conflate sensitivity with fragility. Thornberry addresses this directly in the paper. He argues that this level of sensitivity actually indicates extreme precision within constraint.
Well, let's find a better analogy than just a fragile machine then, because a machine breaks and stays broken. Biology adapts.
Exactly. Don't think of it like a severed wire in a fighter jet where cutting the wire instantly crashes the plane. Think of the genome's context dependency more like a highly complex, self-balancing ecological network, an ecosystem.
Okay, I like that. So taking out a specific regulatory gene is less like cutting a wire and more like removing a keystone predator from a national park.
Exactly.
Like if you take wolves out of Yellowstone, the deer population explodes. The deer eat all the saplings. The riverbanks erode because there are no trees to hold the soil. The rivers literally change course, and the entire physical geography of the park is altered.
That is a phenomenal analogy.
The system isn't fragile. It is deeply constitutionally interdependent. The nodes rely entirely on the context of the whole.
The identity and function of any single part of the genome can only be understood in light of its relation to the whole. And to explain this deep integration, Thornberry actually reaches back to classical metaphysics. He brings in Aristotle and Thomas Aquinas.
Which I have to be honest, seeing ancient Greek philosophy pop up in a paper about modern molecular genomics was a major curveball for me. What do Aristotle and Aquinas have to do with chromatin looping?
It turns out everything. Classical metaphysics wrestled endlessly with the relationship between parts and wholes. Aristotle proposed that physical reality is composed of both matter and form. He called this hylomorphism.
Phylomorphism. Okay.
In Aristotelian terms, form is not just the outward shape of a thing. Form is the internal organizing principle by which matter becomes intelligible and actualized as a specific unified thing.
Okay, we need to ground that in reality. What does form mean in the context of, say, building something?
Let's look at a house. The matter of the house consists of the bricks, the lumber, the copper wiring, the glass. If you dump a pile of bricks and wood on an empty lot, you technically possess all the material causes of a house.
But you do not have a house.
No, you just have a pile of stuff. What makes it a house is the blueprint, the structural organization, the relational arrangement of the parts. That overarching organization is the form. Matter alone, the pile of bricks, cannot account for the structured unity of the system.
And Thomas Aquinas took this concept even further, right?
Yes, he did. The Thomistic development of this idea is that intelligibility, the ability for a system to be rationally understood and to possess a unified purpose, is received through the form, not generated by the matter.
So the individual bricks do not spontaneously generate the idea of a house.
Exactly. The pre-existing idea of the house organizes the bricks.
So what does this all mean when mapping this back to our biology?
Yeah.
The chemical components of our DNA, the carbon, the hydrogen, the nitrogen, the raw nucleotides, that's just the matter, the biological bricks.
Yes.
But the genome, as a complete functioning whole, this vast ecosystem of regulatory elements, long-range chromatin looping, and perfectly timed transcription, that overarching architecture is the form. And Thornberry is arguing that the raw chemical matter doesn't generate that form.
Exactly. Biological information presents itself not merely as raw chemical data reacting blindly, but as formal organization, a principle that actively governs how the material is realized in space and time.
Right.
The genome isn't a list of ingredients. It is the master architect orchestrating how the ingredients relate to one another to create a living organism.
Okay, so we've established that the genome is this incredibly complex, context-dependent, symbolically mapped structure governed by Aristotelian form. But human scientists can only hold so much data in our heads. How did we actually prove the sheer mathematical depth of this order?
We proved it by outsourcing our pattern recognition. Enter artificial intelligence.
Wait. If AI is proving that our DNA is this mathematically pristine, perfectly integrated code, I mean, that completely contradicts what I learned in high school biology. I was taught we are the messy result of random mutations over billions of years. How does random mutation write a flawless mathematical code that a supercomputer can read?
That is the exact friction that Thornberry's paper exploits. The collision of artificial intelligence and genomics is arguably the most philosophically disruptive event in modern science.
Oh, absolutely.
We are talking about massive deep learning systems, large language models, and neural networks developed by places like DeepMind, Stanford, and the Arc Institute.
Well, let's talk about the heavy hitter here, AlphaFold, because the protein folding problem was considered one of the holy grails of biology for half a century. Can you explain why it was such a massive problem and how AI actually solved it?
Gladly. The protein folding problem fundamentally comes down to what is known as Levinthal's paradox. As we discussed earlier, a protein is a long chain of amino acids. To function, it must fold into a very specific three-dimensional shape. In 1969, Cyrus Levinthal calculated that even for a small protein of just 100 amino acids, the number of possible structural conformations is astronomically huge. Something like 10 to the power of 300 possible shapes.
10 to the power of 300. I mean, that's more than the number of atoms in the observable universe.
By a massive margin. It's incomprehensible. Levinthal pointed out that if a protein tried to find its correct functional shape by randomly sampling all possible folds, it would take longer than the age of the universe to fold a single protein.
But that doesn't happen.
No. Inside your cells right now, proteins fold into their exact functional shapes in milliseconds.
So biology knows exactly where it's going. It's not guessing. But for decades, human scientists were guessing. We couldn't look at the linear string of amino acids and predict what 3D shape it would snap into.
Right. Until AlphaFold 2. DeepMind utilized a machine learning architecture called a transformer network, which, by the way, is very similar to the underlying technology of large language models like ChatGPT.
Interesting.
They fed the AI the sequences and known structures of hundreds of thousands of proteins. The AI learned to treat the amino acid sequence like a language. It used spatial graphs to identify the hidden relationships between amino acids that might be far apart on the linear chain, but end up physically touching in the folded 3D structure.
So the AI essentially mapped the grammar and syntax of the biological code, and it worked.
It didn't just work, it completely obliterated the problem. AlphaFold can now predict the 3D structure of almost any protein with atomic-level accuracy simply by reading the linear genetic sequence.
That's wild.
And it goes further. Generative AI models can now hallucinate entirely novel, plausible genetic sequences that satisfy the structural constraints of biology, creating proteins that have never existed in nature but fold perfectly.
Here's where it gets really interesting, though. Here's where Thornberry drops the philosophical hammer. These AI systems, AlphaFold, these massive neural networks, they do not possess understanding. They have zero intentionality.
None. Absolutely zero.
The AI doesn't know what a cell is. It doesn't know what life is. It doesn't know what a human being is. It is a silicon box operating purely on statistical inference, spatial graphing, and incredibly advanced calculus.
Yes. And this raises an important question. If an artificial intelligence that only understands cold mathematics, statistics, and pattern recognition can successfully predict biological behavior with near-perfect accuracy, what does that say about the fundamental nature of biology itself?
It says that biology is fundamentally mathematical. It's compressible. It's inherently rational. Thornberry has this brilliant pivotal phrase in the paper. Pattern recognition presupposes pattern.
That phrase is the absolute heartbeat of this section. The success of AI in modeling the genome is not an artifact of the computer algorithms. The AI is not imposing an artificial mathematical order onto a messy biological reality. The AI is successfully modeling the genome because the genome is inherently ontologically ordered. The capacity of our artificial systems to map genomic structure is entirely parasitic upon the prior existence of that mathematical structure in reality.
It's the ultimate cosmic irony, isn't it? For a century, materialist science tried to reduce life to just messy, random biological accidents. A lucky chemical soup. Then we built these cold silicon brains that only run on pure logic and math. We pointed these silicon brains at our carbon biology to try and sort out the mess.
And the AI came back and said, what mess? This is pristine code. This is a deeply logical, rule-governed system.
The irony is just profound. The ultimate tool of rational calculation has revealed that life itself is rationally calculated. The genome is fully intelligible. It is governed by discoverable mathematical regularities. It is not a chaotic soup.
Not at all.
Okay, so if artificial intelligence proves beyond a shadow of a doubt that the genetic code is mathematically pristine, deeply interdependent, symbolically mapped, and entirely rationally ordered, can our standard, undirected evolutionary models actually explain how it originated?
And now we arrive at the most sensitive, hotly debated, and rigorously analyzed portion of Thornberry's paper, the limits of evolutionary mechanisms. And it is crucial that we look at this impartially, dissecting his distinction between microevolution and macroevolution, strictly following the logic laid out in the source material without falling into ideological traps.
Absolutely. We're looking at this purely through the lens of explanatory sufficiency.
Yeah.
So let's start with what is empirically established, what no one in the scientific community is arguing against, microevolution.
Right. Thornberry makes it emphatically clear that microevolutionary processes are empirically robust and scientifically verified.
We're talking about basic mutation and selection here.
Exactly. We are talking about the mechanisms of genetic mutation, natural selection, and genetic drift. These mechanisms are observable in real time, and they absolutely explain variation, adaptation, and optimization within existing biological systems.
We see this all the time. Bacterial resistance to antibiotics is a prime example. Or, the classic textbook case of the Galapagos finches changing their beak shapes based on food availability during a drought.
A perfect example.
If the environment changes, the organisms with the pre-existing genetic variations that offer a survival advantage will outcompete the others. They survive, pass on those genes, and the population adapts.
Yes. Natural selection is a beautifully elegant mechanism for fine-tuning an existing system to its environment. But the absolutely critical phrase in that sentence is existing system.
Right.
Where Thornberry argues that standard evolutionary mechanisms hit an insurmountable wall of explanatory insufficiency is at the level of ultimate origin. The macroevolutionary question. How did these complex, symbolically encoded, deeply integrated informational systems emerge from non-living chemistry in the first place?
Because optimizing a longer beak to crack a harder seed is fundamentally categorically different than writing the mathematical symbolic code that knows how to build the physical structure of a beak from scratch.
Precisely. Thornberry details several unresolved origins that resist explanation via unguided mechanisms. The foremost is the emergence of the symbolic encoding itself. How did blind chemical reactions invent an arbitrary, rule-based code?
And what's the second?
Second is the establishment of replication fidelity. How did a primitive system learn to copy its code with extreme accuracy without instantly mutating into oblivion? And third, the coordination of the translation machinery.
I'm going to play devil's advocate here because this is where the friction usually happens in these debates. Hasn't the mainstream scientific consensus always been that, given enough time, billions of years, these massive qualitative thresholds are eventually crossed by a billion tiny, unguided quantitative steps?
That has been the assumption, yes.
So why does Thornberry argue that this is a persistent explanatory insufficiency at the level of formal causation, rather than just a temporary gap in our current scientific knowledge? Why can't we just say, we don't know yet, but natural selection will eventually explain it?
It's the essential question, and the answer comes back to the deep mechanical interdependence we discussed earlier. Natural selection, by its very definition, is a filter.
Okay.
It operates on what already exists. It selects for functional advantage. But a filter cannot generate the information it is filtering, and it cannot select for a complex system that hasn't been fully built yet.
Let's ground this in a specific biological mechanism so we can see the paradox clearly. Let's look at the translation machine paradox.
This is the perfect example. To convert the DNA code into the physical proteins that build a living cell, you need an incredibly complex piece of molecular machinery called a ribosome. A ribosome is composed of two major subunits made up of ribosomal RNA and dozens of specific proteins. You also need messenger RNA to carry the code from the DNA and transfer RNA to bring the amino acids.
It's an entire manufacturing plant.
Yes. Now here is the paradox. The DNA contains the instructions to build the ribosome. But the DNA cannot be read and the ribosome cannot be built without a fully functioning ribosome already existing to translate the code.
Oh man, it's the ultimate biological chicken and egg scenario. You absolutely need the translation machine to read the instructions, but the instructions to build the translation machine are locked inside the code that can't be read.
Exactly. Furthermore, both the DNA and the translation machinery require the protective, selectively permeable integrity of a cellular membrane to function.
Right, or they just float away.
Exactly. If they aren't enclosed in a membrane, the chemical components simply diffuse into the environment and dissolve. But the cellular membrane is constructed and maintained by proteins, which are built by the ribosome, which is coded by the DNA.
They all have to exist simultaneously.
They exhibit mutually constrained development. They must exist together as a unified, structurally integrated system to function at all. You cannot build them one tiny, undirected, random step at a time because a half-built ribosome or a half-formed cellular membrane provides absolutely zero survival advantage for natural selection to preserve.
Right. It's useless.
It is biochemically useless until the entire system is complete and closed.
So natural selection is blind. It doesn't look at a half-built ribosome and say, keep going. In a million years, this is going to be incredibly useful. It just discards it.
Exactly. An undirected process cannot logically account for the simultaneous generation of specified, functionally integrated information and the machinery required to decode it. Thornberry is arguing that the mechanism itself, random mutation filtered by selection, is philosophically and structurally inadequate to explain the origin of a symbolic code. Selection does not explain the origin of the formal structures upon which it depends.
Okay. This is the turning point of the deep dive. If classical undirected evolutionary mechanisms hit a brick wall when trying to explain the absolute origin of this incredibly integrated, mathematically pristine, interdependent whole, what is the alternative? Does Thornberry just throw his hands up, declare it a mystery, and walk away?
Far from it. This is where the paper pivots from deconstruction to a highly robust constructive ontology. Thornberry proposes a completely different metaphysical framework through which to view biology, a triadic framework.
And this framework is built on 3 pillars: light, love, and life.
Yes.
Now, I have to admit, when I first read the source material and saw those 3 specific words being used as the foundation of a rigorous academic paper on genomics and artificial intelligence, I paused.
Oh, I'm sure. It's definitely jarring.
Yeah. Using words like light and love in the context of cellular biology sounds jarring. It sounds poetic, perhaps mystical, but not scientific.
It sounds jarring to modern ears because we have been thoroughly conditioned by the language of scientific reductionism. We are used to describing biology exclusively in terms of machines, motors, pathways, and selfish genes.
Very mechanical.
But Thornberry is emphatic that he is not using metaphorical, romantic, or poetic language. He is using these terms as precise, rigid ontological descriptions of biological structure grounded deeply in classical philosophical tradition. So we need to break them down carefully, starting with light.
Let's unpack the first pillar. Thornberry defines light as intelligibility.
Yes. Light, in its classical philosophical sense, dating back to Plato and Augustine, is the fundamental condition of knowability.
Okay, what does that mean in practice?
Consider physical light. Physical light illuminates matter, making physical objects visible to the physical eye. Ontological light, therefore, is the principle that illuminates the structure of reality, making it visible to the rational intellect. A system possesses light insofar as it exhibits a logical structure that is inherently accessible to rational inquiry.
And as we just discussed with the AI models, the human genome is profoundly, astonishingly intelligible.
Exactly. The genome can be mapped. Its dependencies can be graphed, its mathematical regularities can be discovered and predicted. When a geneticist looks through an electron microscope, or when a neural network analyzes a multi-terabyte dataset of amino acid sequences, they are not imposing a human or artificial order onto biological chaos.
They're just reading what's already there.
They are encountering an order that pre-exists their observation. That inherent availability to rational understanding, that mathematical grammar, is what Thornberry rigorously defines as light.
It's the profound realization that the physical universe possesses a rational syntax that our minds and our machines can actually read.
Yeah.
That makes total sense to me. Okay, let's move to the second pillar, which is the one that really challenged my traditional view of biology.
Right.
Love. Thornberry defines love as relational coherence. I really want you to deeply analyze this for us, because redefining biological interdependence as love, an ontological drive toward relational unity, how does that tangibly change how a scientist looks at a living physical system?
To grasp this, we have to entirely strip away our modern romanticized emotional definition of love.
No Valentine's Day cards in the lab.
Right, no romance. Thornberry is utilizing love in its profound ancient ontological sense. Love is the active principle by which multiplicity is integrated into unity. It is the binding force of relation that makes a whole greater than the sum of its parts.
Okay.
If you look at the architecture of biological systems through this lens, what do you physically see? You see individual organelles cooperating to sustain a cell. You see millions of cells communicating to form tissues. You see organs functioning not for their own isolated survival, but for the life of the whole body. You see those incredibly complex, long-range chromatin loops we discussed, operating in perfectly coordinated relation to transcribe a single protein.
So a single cardiac muscle cell in my heart has no intrinsic meaning, no ultimate purpose, and frankly cannot survive without the context of the lung cell providing oxygen, the blood vessel providing transport, and the neurological electrical impulse telling it when to contract.
Exactly. No single component of a living biological system is fully intelligible in isolation. The identity of the part is entirely defined by its internal relations to the whole. The unity of the organism isn't just an accident of molecules bumping into each other in a confined space. It is a constitutive driving reality. That profound drive toward relational coherence, toward the integration of disparate matter into a unified functioning organism. That is what Thornberry calls love.
That is heavy. So when a scientist looks through a microscope with this ontological framework, they don't just see a wet machine made of independent, selfish genetic cogs fighting for survival. They see a vast community of matter, bound together by a fundamental physical principle of relational unity. It shifts biology from a science of isolation to a science of profound connection.
And if we connect this to the bigger picture of the paper, it demonstrates how classical philosophy and cutting-edge genomics are arriving at the exact same conclusion from opposite directions.
Oh, that's a great point.
Artificial intelligence proves that the genome is an inextricably linked web of deep dependencies. Classical philosophy tells us that the ultimate principle binding a web of dependencies into a single cohesive organism is relational coherence or love.
Which brings us to the third and final pillar. Life. Thornberry defines life as actualization.
Yes. Life is not mere random molecular activity. It is ordered activity directed specifically toward the maintenance of coherence.
Okay, unpack that a bit.
Think about what living biological systems actually do on a microscopic level. They maintain homeostasis, internal stability against environmental chaos. They actively heal and repair damaged DNA. They reproduce. They respond adaptively to their environment, but always within the specific structural constraints of their form.
So they are actively expressing the light and love.
Precisely. Thornberry relates this directly to Aristotle's concept of the soul. For Aristotle, the soul was not some ethereal, ghostly vapor haunting a biological machine.
Right.
The soul was the form of a living body. It was the organizing principle by which a body is actually alive rather than merely assembled. Life, in Thornberry's triadic framework, is the dynamic physical realization, the actualization of that encoded order.
So to synthesize this immense philosophical framework for a moment, the genome encodes light, the rational, intelligible mathematical structure. It coordinates love, the profound relational unity and contextual interdependence of all the biological parts. And it enables life, the dynamic physical actualization of that integrated order over time. Light, love, and life, a hierarchy of being.
And it is vital to remember that this triad is presented not as a poetic overlay to make us feel good about biology, but as a rigid, accurate description of the ontological structure of biological reality, as it actually presents itself to us through modern genomic science.
Understanding our biology as this profoundly integrated architecture of light, love, and life is a breathtaking paradigm shift, but we are no longer just observing biology. We are actively altering it.
We are indeed.
You, listening right now, you are living in the era of CRISPR-Cas9. You are living in the age of gene editing, of synthetic biology, of human-directed evolution. And that brings us to the final and perhaps most urgent section of Thornberry's paper, human knowledge and ethical constraint.
This is where the metaphysical framework crashes headlong into the hard reality of modern laboratory science. Because despite our incredible technological advances, despite AlphaFold and CRISPR-Thornberry issues a profound reminder that human agency remains strictly bounded.
Bounded how?
Well, we have the power to modify genomes, but we do not have the power to originate them. We can use AI to model biological outcomes, but we cannot fully predict the cascading systemic effects. We can manipulate these complex systems, but we absolutely cannot recreate their totality from scratch.
To use a literary term, we are incredibly skilled editors, but we are not the authors. We are tinkering with a machine we did not invent, and whose deepest grammar we are only just beginning to map.
Exactly. We operate entirely within an order we did not establish, and that realization carries immense, unavoidable ethical weight. If biological systems embody this intrinsic mathematical intelligibility, light, and this profound, fragile, relational coherence love, then they are not just neutral substrates. They aren't just putty for us to mold however we see fit for industrial or medical convenience.
They aren't a random assortment of biological Lego bricks that we can just snap apart, swap around, and plug back together without consequences.
No. Intervention in a system this profoundly integrated requires vastly more than just technical mechanical competence. It requires ontological awareness. You have to understand the fundamental nature of the thing you are editing.
Good.
To illustrate this, Thornberry uses a fascinating historical analogy drawing on the 19th century art critic and polymath John Ruskin, specifically referencing Ruskin's The Seven Lamps of Architecture.
Yes. I loved this application. Ruskin argued that true craftsmanship, particularly in grand architecture, demands a fundamental respect for the nature of the material. You must honor stone as stone, and wood is wood.
Precisely.
You don't try to force stone to do something it structurally wasn't meant to do. You don't treat it like iron, or you inevitably compromise the integrity and the beauty of the entire building.
And Thornberry argues that in the realm of synthetic biology, this principle of structural respect applies with exponentially greater force. When geneticists go in to tweak a gene using CRISPR, they aren't just making a localized mechanical repair. They're intervening in a profoundly integrated, extraordinarily sensitive architecture.
Right, because of all those long-range dependencies we talked about.
Exactly. If you do not respect the material, if you do not respect the deep relational coherence, the topologically associating domains, the long-range dependencies of the genome, your technical intervention might cause catastrophic systemic failure. We see this with off-target effects in gene editing, where changing a sequence in one place inadvertently causes a dangerous mutation thousands of base pairs away.
It's the difference between a mechanic swapping out a faulty spark plug in a car and an editor trying to change a pivotal word in a Shakespearean sonnet. The car is an aggregation of modular parts. The sonnet is an integrated formal whole where changing a single word alters the rhythm, the rhyme, the meter, and the meaning of the entire poem.
That's beautifully put.
When we use synthetic biology to edit a genome, Thornberry is arguing we are editing a sonnet, not a spark plug. And if we lack the ontological awareness of the love, the relational coherence connecting that single gene to thousands of others in the biological ecosystem, we're acting with dangerous blind hubris.
It is a profound, timely warning against reductionist thinking in the age of genetic engineering. Biology, at its deepest ontological level, is not explained by reduction to its lowest parts. It is disclosed through the profound order of the whole.
So what does this all mean for us as we synthesize this incredible mind-bending journey we've been on today? We started by opening the door to the human genome, expecting to find a messy, random attic of accumulated evolutionary junk. Instead, we found an astonishingly precise, tightly regulated reference library.
We certainly did.
We discovered that DNA is a true symbolic code governed by arbitrary rules, not just blind, inevitable chemistry. We saw how the immense, context-dependent, three-dimensionally folded web of the genome demands an understanding of classical Aristotelian form.
And we explored how the explosive application of artificial intelligence to genomics has not dissolved the mystery of life, but rather clarified its astonishing structural reality. The AI revealed a mathematically pristine code, proving definitively that pattern recognition presupposes pattern, and this massive revelation forces us to acknowledge that while microevolutionary processes brilliantly explain local adaptation and variation, they fall catastrophically short of explaining the ultimate origin of this formal, symbolic, interdependent architecture.
Which ultimately led us to Thornberry's magnificent triadic framework life as ordered light. The physical embodiment of intelligibility, relational coherence, and actualization. Light, love, and life.
The ultimate takeaway from Thornberry's work is a necessary reorientation of our scientific and philosophical posture. The primary task before both modern science and philosophy is not to artificially diminish the profound order of the genome by forcing it into reductionist, materialist models that clearly no longer fit the data. The task is to acknowledge that order, to respect its coherence, and to understand it more fully with a profound sense of humility and awe.
Humility and awe. That is exactly the right note to end on. But as we wrap up this deep dive, I want to leave you listening with a final provocative thought to mull over, something that builds on Thornberry's foundational work, stretching into the very near, potentially unsettling future.
Ooh, I like where this is going.
We talked about how human beings currently only possess the bounded power to modify this intricate genetic code rather than originate it. We are editors of the biological sonnet, But what happens when we eventually cross that technological Rubicon? What happens when we ask the AI, the very tool that helped us read the mathematical light of the genome, to write entirely new synthetic forms of biological information completely from scratch?
That is the million-dollar question.
If an artificial intelligence, a silicon entity, which possesses absolutely zero intentionality, zero consciousness, and zero understanding of what it means to be alive, originates a massive novel biological sequence, will that synthetic organism inherently possess the love, that deep constitutive relational coherence, and the true life that Thornberry describes as the soul of biological reality? Or will we have simply used advanced calculus to create a highly functional but ontologically empty mimicry of biology? Will that synthetic creation be a true addition to the grand biological reference library? Or just a perfectly bound, mathematically flawless book with entirely blank pages? Thank you for joining us on this deep dive. Keep questioning the structure of the reality around you, and we'll see you next time.